with structural colors. So of course, color is an important mediator of fitness. And in addition to color that's derived from pigments like melanin or chlorophyll, there's also a whole category called structural color, which is derived essentially from morphology. So this is cases where there are nanostructures, their size is on the order of the wavelength of light, they're near the surface of the organism, and they cause interference of light that produces a color. Now, unlike pigmentary color, structural color can cause not only a hue, but also a variety of optical effects like iridescence or polarized light. It's a pervasive phenomenon, and it's also really beautiful. So to explain just a bit more explicitly how it works mechanistically, I want you to imagine the simplest possible example of a structural color, which is just a simple plane or a sheet of some material that's a high refractive index. Here we'll consider chitin. And so what happens is that when light encounters this boundary between the two different refractive indices, it changes speed, bends, and it also produces a reflection. That all happens again at the other boundary. And so then at the end of this process, we have two different reflections that have the opportunity to interfere with each other. They may or may not still be in phase. And whether they're in phase depends on how far extra this second reflection has traveled in the medium and whether that's a multiple of its own wavelength. So if it is a multiple, then these two reflections will still be in phase, they'll interfere constructively and produce an observable color. And so this is all wavelength dependent, of course. So for any particular thickness of this medium, only some wavelengths will still be constructively interfering at the end. The same principle applies for more complex morphologies. So I'm studying structural color using a butterfly model. This is the Buckeye butterfly, or Junonia acenia. And it's definitely not the butterfly you would pick as a showy, iridescent, structural colored example. However, it is a pretty good lab model. And I was drawn to this system because about the time that I was starting grad school, there was a private butterfly breeder in Florida who noticed that sometimes her animals had a little sprinkle of a few blue scales right up here on the forewing, and she began selectively breeding those to artificially select for blue. And like I say, when I was starting my project, she was advertising Buckeyes like this, which was very exciting to me because blue pigments are so rare that this is almost an immediate giveaway that she's somehow created a structural color by artificial selection. So she describes that process as taking about a year. That's like 6 to 12-ish maybe butterfly generations. And so the first research question that I took on was trying to identify what changed structurally between these and wild type scales, and then whether there were also concordant differences in the pigmentation. So let me just orient you a little bit to butterfly scales and how they work. So the pattern on a butterfly wing is a mosaic of a lot of different scales. And if you look at any one, it looks something like this. They're about 300 microns long. In an adult wing, they're a dead epithelial structure. They're made completely of chitin. They insert into a socket on the wing membrane over here. And this scale, I've cut a hole with an ion beam so that you can see some of the finer architecture that they have. So this is a typical default unspecialized scale. And they have a sheet of chitin that extends the whole surface of the scale. This is called the lamina that you can see here, and this will be important later in the talk. And this supports all these other elements. So we have ridges, we have cross ribs, micro ribs you may be able to slightly see in this image. And you can find examples of different butterfly taxa that have elaborated any number of these elements or altered the spacing a little bit to create various kinds of really showy structural colors. So here's my blue scales, and you can see this is the top view. You can somewhat see the ridges I just pointed out to you. This is the flip side, so you're looking at just the smooth lamina. I can formally prove that yes, this is a structural color by immersing the whole thing in an indexed matched fluid. So that, of course, eliminates the boundaries between different refractive indices. There's no opportunity for refraction, and so anything you can see in these conditions is a pigment. So here we go. Of course, it's no longer blue, so the blue is structural, but there is also a little bit of a dark pigment that's mostly localized in the ridges. And then the other thing that I can show you is that I can dissect off the ridges and ribs, and you can see that just bare lamina is sufficient to have a blue structural color. So I was excited because in the literature there's examples of butterflies where rather than having just that simple plain lamina, they've elaborated it to be a multi-layer or to have pockets full of a honeycomb structure, things like this that make some pretty fancy structural colors. So the next thing that I'm going to show you then is a cross-section through the scale this way, and then looking down onto the cut edge with electron microscopy to see what features there are. So here's my blue scale cross-sectioned. And when I first saw this, I was kind of dismayed because it looks the same as the default scale to me. There's certainly no honeycombs or multi-layers there. However, if we measure the thickness of the lamina, so that's this thickness here, it's 210 nanometers thick. And actually that is the right thickness to make blue just as a simple thin film reflector, which is that simplest case I explained at the beginning. So how is that different structurally from wild type, or is it just simply a loss of a dark pigment? Here's the wild type scales, and you can see for all that they're brown, actually wild type scales have the same amount of pigment as my blue scales, and you can get a hint already from the colors here and here that there's something structurally different about the lamina. So when I cross-section these, here we are. Here's the thickness of the lamina, and it's dramatically thinner now. It's only about 140 nanometers thick. So what this means then, the first result I'm showing you is that butterflies respond rapidly to direct selection on color by increasing the thickness of their scale laminas by about 50%. And that was really interesting to me because, first of all, it's like the default expectation, even the simplest butterfly scale has to have a lamina. And so if that can be a thin film reflector, and if even in brown animals there's some structural gold contribution to the overall color, then maybe we should be thinking of structural color as kind of a default that it virtually always contributes at least a little bit to butterfly coloration. The other thing that was an intriguing possibility to me is that this suggests that perhaps it's not so difficult for them to change the thickness of the lamina. And you can imagine hypothetically that if you can produce any thickness, you could produce any color, all with this same trick. So I was curious, is that something that they do? If so, that fits well with this modeling that was done by Stavenga's lab group. So this is just showing you hypothetical reflectance curves like you would measure from a spectrophotometer off of planes of chitin of increasing thickness. So if we look at the one that's about the thickness of my blue scales, it makes sense. In the visible part of the spectrum, there's just one peak, and it's in blue. If we look at the one that's similar to my gold scales, there is no one peak in the visible part of the spectrum. However, there's a lot of reflectance in all the long wavelengths, so it makes sense that your eye would interpret that as gold. But again, the thing I want you to notice about this is that there's kind of a typical shape of a reflectance curve off of a thin film. And as we increase thickness, this stretches towards the longer wavelengths. So again, you can imagine that if you could tune this perfectly, you could perhaps produce any color just with a thin film. So to investigate whether this is in fact something that butterflies routinely do, I characterized scales from many different species of color morphs in the larger genus. So at the top here we have the species that I work on, Senia, and you can see again natural and artificial selection that I've been showing you. But of course you can also see that there is a lot of color variation in this genus, and the way that it's partitioned suggests that it's likely functional in this group, and it probably responds to multiple types of selective drivers. So here we have geographic variation of brown and blue. We have a whole clade where males always have a showy blue patch and females have reduced or absent blue. And then we also have an example where the animals are blue or red depending the season. So I won't drag you through all the crosswise scale comparisons, but suffice it to say, yes, they certainly can make many different thicknesses of colors, and there's always a thin film that's contributing somehow to the overall color. So for instance, we have a number of comparisons of brown to blue, and those are exactly the same thing that's happening in my Buckeyes that were artificially selected. But what might surprise you is that even some of these other colors, like oranges and yellows, that you would think could easily be produced by a pigment, they color match. So they'll have a yellow or orange pigment, but they'll also have a yellow or orange thin film as well, reinforcing that. And then, for example, these two species, they're just kind of buff and beige. You would not think of them as structurally colored. They do have a little bit of a pigmentation difference, but actually, I think more important is the difference in the thickness in their laminas as well. So yes, this seems like this is an important and common tool that they use in their coloration kit. Let me show you kind of the same point, but from another angle. This is really cool. This is a video taken by my lab mate Aaron, and he's prepped a butterfly pupa so that you can watch the wing development live. And as it starts to get silvery, that's when chitin's being deposited. And then the scales continue to develop, and right at the end of development, all the pigments get deposited here. So you can see this is a butterfly from my line, and it's going to end up with kind of turquoise scales in this part of the wing. So I'm going to play it again, zoomed in there, just starting when chitin's first starting to be deposited. And I want you to watch this region. You're going to see it shift like a rainbow in exactly the order that we would predict. As thickness increases, the color would shift. So we go, maybe that went too fast. I'll play one more time. We start kind of gold, purple, and then blue, and then increasingly greener and greener until it's turquoise. So again, this suggests that chitin gets deposited gradually. It's not sort of you can have X or 2X units of chitin thickness, but that developmentally as well, probably any thickness or therefore any color is possible. Okay, so I'll talk briefly about this. This is ongoing, but I'm also interested in genetically mapping what loci control this color tool. And so this is a cross between an artificially selected blue and a brown lab line animal. Give you a quick peek at some of my F2s. You can see that there's variation for how much of the wing has structurally colored scales, but once again we have different colors popping out, not only blue but green and indigo. And I can confirm with SEM that these additional colors represent additional thicknesses like you would predict in the model. So I'm interested in mapping hue. I'm also interested in mapping what percent of the wing has modified color scales on it. Just to summarize, so I've shown you that scale laminas in butterflies are a thin film reflector, that they respond to selection on color, they're tunable, and they can make many colors with the same mechanism. And I've shown you that hopefully soon we'll know something about the genetic architecture of this trait. So thank you. Is there any question?
Yes. Do you think that this is a more widespread mechanism among butterflies, apart from the ones you've studied, or?
I think that it probably is. There's a paper in another group of nymphalid butterflies where this is reported. And like I said, this is kind of the simplest possible scale architecture, so yes, I think it's probably very common.